What's going on, Science 10s? It's Mr. Walton here. Today we're talking about active transport. So far, we've only really talked about particles moving with their concentration gradient, or we call down their concentration gradient. What we mean by that is we mean from a high concentration to a low concentration. Remember, if we've got some sort of a beaker or container and we put some particles in one corner of that container, the particles are going to naturally, because they're all moving in random directions, these particles are naturally going to spread out. They're going to move from an area of high concentration, that's an H, to areas of low concentration. And that doesn't take any energy. We don't need any additional energy to be able to do that. But okay, so this helps this helps even out the concentrations of particles inside and outside of the cell, right? It's going to help kind of spread out those particles. However, there are many examples of substances that constantly need to be moved against their concentration gradient, either being stored in high concentration inside or outside of the cell. And let's take a look at a very specific example of something. So in your stomach, there are specialized cells that they absorb nutrients. And so, for example, these cells, they want to store glucose. They want to be able to absorb glucose out of your small intestine. So in the example above, glucose is in high concentration inside this cell. So we've got an area of high concentration for glucose. There's low concentration of glucose outside of the cell. Cells need a constant supply of glucose, and if the membrane was permeable to glucose, let's just think about this for a second. If this membrane was permeable to glucose, what would happen to the glucose? Well, yeah, maybe a couple of these would move inside the cell, right? They're all moving in random directions. So maybe a couple of these would move inside the cell, but many of these would move outside of the cell. So if we just let the glucose, or if we let the membrane be permeable to glucose, then we would have a net flow out of the cell by the glucose. And that's not what we want. We want the glucose to be stored inside the cell. So there would be a net flow out of the cell, which is not a good thing. The cell needs a way to be able to pump the glucose molecules against its concentration gradient. This requires the use of ATP. So what we want to do is we want to take these glucose molecules and pump them inside the cell. Well, in order to do that, we have to be able to use up ATP. And ATP just stands for adenosine triphosphate. Think of it like, if you want to go out and you want to do something, you have to pay money in order to do it. Well, the chemical currency inside the cell is ATP. You have to pay ATP. That's the energy that causes things to happen in cells. So let's take a closer look at what adenosine triphosphate is. So ATP is adenosine triphosphate. And you can see the structure over on the side here. It's this thing called adenosine, which is really just, it's two different substances bonded together. It's adenine and then a sugar, a ribose backbone. So there's this adenosine, and then there's 1, 2, 3 phosphates. So we call it adenosine triphosphate. Adenosine triphosphate, oh man, I'm struggling here, is a molecule that is used whenever the cell needs to accomplish a task. One of the phosphate groups here, the third phosphate, breaks away and energy is released. A large amount of energy can be released so that the cell can complete those tasks. Now, do we need ATP to be able to force things to go against their concentration gradient? Yeah, we do. because otherwise, if we don't put energy in, then these things start evening out, right? Everything wants to be a uniform concentration inside and out. So in order for us to store things in high concentration, we need to use up ATP. In general, there are two examples that we need to know for Science 10 of active transport. The first one is the sodium-potassium pump. So, very important cellular pump in nerve cells. Your nerve cells wouldn't work without it. It pumps sodium and potassium ions against their concentration gradient using ATP, and it keeps a high concentration of sodium out of the cell and a high concentration of potassium inside the cell. So what happens here is that we've got our cell membrane, and we've got a protein. This is a protein channel, or a protein carrier, I guess we should say. And so the sodium inside the cell binds to here, and then an ATP comes in and it adds a phosphate group to the protein, and that changes the shape of the protein. Now that's pretty crazy. You've never seen something like that before, but that's what happens is that when the energy is released from the ATP, that phosphate group causes a change in the shape of the protein, and these sodium groups get kicked out. So they bind on the inside of the protein, and then when it changes shape, they get kicked out, and then now potassium can bind, and then it can flow in. So you don't actually have to know any specifics about this sodium-potassium pump. All you need to know is right here. So you need to know that it pumps sodium and potassium ions against their concentration gradient, and you need to know that it uses ATP. So I suppose, really, that's the only piece of information that you need, and it uses a protein carrier or a protein channel. So that's the sodium-potassium pump. The second example of active transport that we need to know is endocytosis and exocytosis. And it's a lot like endothermic and exothermic in that endo, you can think of that as like enter the cytoplasm or the cell, and then exo means exit. So this is just active transport of substances into or out of the cell via vesicle formation. Now, a vesicle, remember, is just a membrane-bound organelle. So what's happening down here, this is exocytosis. So let's say we have the inside of the cell and then the outside of the cell. So we've got a bunch of waste or something that we want to get rid of. It doesn't always have to be waste. Sometimes it's neurotransmitters in our brain. This is how you send chemical signals from one nerve to another inside your brain. And so we've got a vesicle. Remember, a vesicle is just a vehicle to be able to move things around. A vesicle with a bunch of stuff inside of it. And that vesicle ends up making its way towards the membrane, and the vesicle and the membrane fuse together, and they release all of these particles. So these particles are exiting, and so we call it exocytosis. So this process does indeed take ATP. It requires energy to be able to do this. So it's a form of active transport. So that's exocytosis, and hopefully it won't be too confusing that endocytosis is basically the exact opposite of this. So here, I'm actually going to draw a little bit different of a picture. Okay, so let's say I have a cell, and there's something that the cell wants to engulf. It wants to take in this thing. What's going to happen is this cell will kind of reach around this object. And so the cell will reach around this object, and it will try to engulf this object. And it'll form a vesicle. And so this is endocytosis, because this object is entering the cell. Perfect. The process that I drew here is specifically called phagocytosis. So phagocytosis is the taking in of a solid by the formation of a vesicle. So it's the cell eating. May involve receptors on the surface of the cell membrane to initiate. Yeah, okay, sure. So this cell, when it bumps into this thing, it bumps into it, and then it says, oh, right, that's the thing that I want to eat. So it engulfs around it, and then it can form a vesicle. The cell can do the exact same thing if it wants to take up a liquid or a substance dissolved in the liquid by something called pinocytosis. So some people call it pinocytosis. I call it pinocytosis because it reminds me of the word pint. A pint is a volume of liquid. You could go order a pint of water, or you could go order a pint of a drink or something like that at a restaurant. And so pint means liquid to me. So that's why I call it pinocytosis. It's the cell basically drinking in something. So those are in general, the two basic types of active transport that you need to be able to know. You need to be able to know the sodium-potassium pump, only that a protein channel or a protein carrier is required, and it pumps sodium and potassium ions against their concentration gradient using ATP. And then also endocytosis and exocytosis. Now, endocytosis and exocytosis are actually really quite simple. It's just the cell either eating something or drinking something, or, right, another way to think about exocytosis is like the cell, this is going to be gross, but the cell puking up something, right? So the cell has a vacuole in it, and the cell wants to get rid of it. So this thing's going to start moving towards the edge. This vesicle is going to start moving towards the edge, and then they will bind with each other, and this thing will be able to get released. So this is exocytosis. Again, it's just whatever the cell wants to either get rid of or release to its surroundings. So exocytosis and endocytosis. Let us know if you have any questions. Talk to us in the office hours, right? Or email us if you want to get into contact with us. So otherwise, I don't know. Have a great day.